This is A. Mandri Kumar Rajesh. In this video, we are going to discuss a topic called isolation of DNA material with the help of gel electrophoresis. What is meant by gel electrophoresis? So, it is a novel technique used to isolate the DNA molecule or RNA molecule or even a protein molecule can be isolated based on their size when they are subjected into the electric field. And that technique is called as gel electrophoresis. So, gel means in this technique we are using a gelatin substance. We are using a gelatin substance. Then we are using electro field, that is the electric field. So that is why they are mentioned electro. So here we are using electric field. Then phoresis. Phoresis means movement. Phoresis means movement. So when we applying the electric field, the movement of the molecule takes place. That is why it is mentioned as gel electrophoresis. Then some of the factors which can influence the rate of migration, that is the movement of molecule, and that factors are the net charge of the molecule, whether that molecule may be a DNA or RNA or a protein. So the rate of migration depends upon the net charge of the molecule, net charges of the molecule. The next point is, it depends upon, the migration depends upon the size of that molecule. Migration depends upon size of that molecule. In addition, it depends upon ionic strength of the buffer solution. Ionic strength of the buffer solution. Then the fourth point, it depends upon the viscosity of the buffer. Viscosity of the buffer solution. The fifth point is temperature of the medium. Temperature of the medium. So these are the factors which can influence the rate of migration or the movement of the molecule. So net charges of the molecule, size of that molecule, ionic strength of the buffer solution, then viscosity of the buffer solution, and temperature of the medium. So these are the important factors which can influence the rate of migration. Then let me tell you the principle of gel electrophoresis. Suppose that molecule may be a positively charged molecule. When they are subjected to electric field, it can move towards the negatively charged terminal. So if that molecule is positively charged cation, when they are subjected to electric field, it can move towards negative cathode, a negative terminal. Suppose if it is a negatively charged molecule, so it is maybe an anion. When they are subjected to electric field, it can move towards positive anode or positive terminal. Suppose if it is a DNA molecule, when the DNA molecules are subjected to electric field, they can move towards the positive anode because DNA is negatively charged. So DNA is negatively charged molecule. Negatively charged molecule due to the presence of phosphate groups of the sugar, due to the presence of phosphate groups of the sugars and phosphate backbones So due to the presence of phosphate groups of the sugar and phosphate backbones, the DNA is negatively charged. So when it is subjected to electric field, it moves towards the positive anode. Then the movement of DNA will be directly proportional to the phosphate group in it. The movement of DNA will be directly proportional to the phosphate groups in DNA. So directly proportional to the number of phosphate groups in DNA. Then there are two types of gel electrophoresis. They are Agarose gel electrophoresis and Polyacrylamide gel electrophoresis. I give you the difference between the two gel electrophoresis. Let me point out the difference between Agarose gel electrophoresis and Polyacrylamide gel electrophoresis. Here I have written three major differences between these two electrophoresis. In Agarose gel electrophoresis, the Agarose gel is entirely non-toxic. They are not harmful to us. While in the case of Polyacrylamide gel, it is moderately hazardous and it requires protection during handling. So it is moderately poisonous. So we have to make precaution measures before we handling the polyacrylamide gel. Agarose gel is complex gel because it is made up of many differently sized molecules and it has wider gaps. It has wide gaps. Agarose gel is a complex gel because it is made up of many differently sized molecules and it has wide gaps. While in the case of polyacrylamide gel, it is made up of only one large molecule and it has smaller gaps. Agarose is poured horizontally. In the Agarose gel electrophoresis, the Agarose gel has to pour horizontally. While in the case of polyacrylamide gel electrophoresis, the gel has to pour vertically. Then it is a structure of horizontally gel electrophoresis units shows the buffer reservoir, positive electrode, negative electrode, gel platform and cover. Then I am going to tell you the steps which are involved in the Agarose gel electrophoresis. Let me explain the steps which are involved in Agarose gel electrophoresis. Before that, I would like to give you a brief introduction. Actually, Agarose gel electrophoresis is otherwise called as submarine electrophoresis or horizontal gel electrophoresis. So it is otherwise called as submarine gel electrophoresis or horizontal gel electrophoresis. You already know why it is otherwise called as horizontal gel electrophoresis, because that Agarose gel has to pour horizontally. Then why it is otherwise called as submarine gel electrophoresis? I will give you the reason within a few minutes. Then the Agarose powder is a purified polysaccharide which is derived from the red algae Gelidium and Gracilaria. Actually, it is a purified polysaccharide which is derived from the red algae Gelidium and Gracilaria. Agarose can dissolve by boiling in buffer solution and it allowed to cool. This results in the formation of Agarose gel at room temperature. So that Agarose powder can be dissolved in the boiling buffer and it allowed to cool. This results in the formation of Agarose gel at room temperature. Then Agarose gels are most convenient to separate DNA from few hundred base pairs. So the Agarose gels are more convenient to separate DNA from few hundred base pairs and onwards. So that's all about the introduction part. Let us talk about the steps which are involved in the Agarose gel electrophoresis. So the DNA of varying length is mixed with sucrose or glycerol and tracking dye. So tracking dye must be ethidium bromide because here we are going to isolate the DNA molecule. So we have to use ethidium bromide. Then this sucrose increases the density of the DNA sample. So due to increase in the density of DNA sample, it sinks to the bottom of the well. That is why the Agarose gel electrophoresis is otherwise called as submarine gel electrophoresis. Due to the increase in the density of DNA sample, it sinks. The reason is sucrose. The sucrose increases the density of the DNA sample. Then one of the well is loaded with standard marker DNA of known length. So this marker DNA are used to compare with unknown DNA fragments. The next step, electric current is applied to the electrophoresis. So the electric current is applied to the electrophoresis unit between 50 to 150 watts. 50 to 150 watts. So the DNA loading dye migrates out of the well and enters the gel respect to the electric field. So the DNA loading dye migrates out of the wall, and it enters into the gel plate in respect to the electric field. Then the moving of the DNA is monitored by observing the dye in front. So due to that electric field, the dyes migrate. So along with the dye, DNA also migrate. So we can monitor the moving of DNA by observing the dye in front. Then the electrophoresis is halted when the tracking dye is at another end of the gel. So we can stop that process when the tracking dye is at another end of the gel. Then we are able to get bands of DNA. On the gel plate, and the gel plate are eliminated under the UV rays. Then with the help of that ethidium bromide state, that bands illuminates orange fluorescence. The bands illuminates orange fluorescence. Then we can take a photograph. So this is how we have to isolate a DNA molecule. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.